Now we will welcome one of my colleagues. Raj is technical director for Americas, and Raj will discuss about the topic which is a bit contradictory sometimes on the field about subclinical damage in the gut.
Thank you, Mikhail. All right. So some of the points that were already covered with Siska and Bob, and I would like to go on a different tangent and to look on some of the biomarkers that we can evaluate to look for subclinical challenges. And I'll present some data and some of the alternative approaches on the subclinical challenges and how we can handle them. Before going into that, I would like to kind of give a brief overview on why do we need to look for that. And so how far these subclinical challenges are, I mean, it's obvious, we tend to lose performance based on these challenges, but how far these affect on a day-to-day basis. As we see, the dietary energy comprises of 45 to 50% of poultry production cost, either in broiler or layer production. But the gut consumes around 20% of the nutrients absorbed from the diet. But on a challenged gut, which is has a compromised epithelial integrity, increases the energy expenditure to 45%, which is almost half of the energy or nutrients absorbed from the diet goes for the maintenance or repair of the gut epithelium. And then these subclinical challenges, they constantly divert, this is an ongoing basis, about 10 to 12% of dietary nutrients towards the activation of immune system. Because as soon as there is an infection or there is a challenge, then the body or the host starts the immune system to go and rectify the inflammatory effects that are caused by the the challenge. So the animal tends to spend some amount of energy, which is about 10 to 12%, and this has been proven by Dr. Klasing's group over the years. This amount of nutrient loss by the animal on a constant basis. So this is quite obvious that these challenges compromise the gut integrity and create a loss in the productivity. So when we look for how this happens, this is a brief overview on, it starts from a stress, and looking at a stress, it could be any type of stress. It could be pathogenic, environment, heat stress, high humidity, and management, particularly like vaccinations, transportation, and raising birds in built-up litter, and feed withdrawal, and fasting, feed change. So even we think from changing from starter to grower, because it's a normal process, but again, that change also provides a challenge to the bird. And the bird has to go through this challenge in order to increase the productivity. So what happens is this stress provides an opportunity for the subclinical challenge. It could be, again, through any of the organisms that have been already discussed by the previous speakers, mycotoxins or any of the pathogen. And they compromise the gut integrity, and that leads to constant activation of immune system, leading to increased energy expenditure and drop in performance. So looking at the subclinical challenges, here you can see two images. This is quite old experiment, and it was published one. And on the left-hand side here, you can see the birds raised on a new quarter. So that means a new litter. And this is a basal diet on black dots, and then the white dots were diets the birds fed with virginiamycin as an AGP. And there's no change in the performance. But again, on the same experiment, two other groups, and then an introduction of contamination was done after the first week, and here you can see the drop in performance with the basal diet, and then the birds that were fed the virginiamycin, which is the antibiotic, didn't lose that performance. So basically, this is the basic idea why we were feeding AGPs throughout the life of the chicken, just to maintain this growth curve. So now the situation is different with challenges of using antibiotics in the diet, and that opens up a big Pandora box of what should we do if we are without antibiotics and if we go back to this situation. And this is on a normal basis. And this is another experiment. Here again, 4 different treatment groups, a control, and this is a Emeria tenella challenge group, and this is Salmonella typhimurium, and this is again a combination of both. 7, 10, and 14 days after the challenge, the control group had no Salmonella. The Emeria group obviously was not challenged with Salmonella, so there's no Salmonella. And here you can see the Salmonella group had, of course, it was challenged, so it had some Salmonella. But the difference here, the last group, which was again exposed to both challenges, the cocci and Salmonella, had increased amounts of Salmonella. So this is a potentiation of one pathogen to other pathogen. So if you have already a cocci that is present, that predisposes the bird to another potential infection, and that increases the impact of this infection here. And this is another case where Emeria maxima, Clostridium perfringens, and here you can see Emeria maxima did some reduce some body weight. Clostridium didn't have when it was challenged alone, but when the combination had a higher effect on body weight gain. Again, the same result repeated in lesion scoring. And here you can see the livability, which is on the opposite of mortality. So the number of birds that are alive were about 89 to 90% with the combination, whereas the individual challenges didn't do. Of course, they did some damage with the Clostridium, but there was an additive effect with the combination. And again, this was one of the experiments that we did. Here you can see the control group and the cocci challenge group. No difference in feed intake, body weight gain. Yes, there was some drop in body weight gain and FCR. But the interesting thing was, these were challenged with Emeria species, but there were no absolute clinical symptoms. These birds were just quite normal, even though they were challenged with coccidiosis. But again, the loss was the performance. So this is the point when we talk about subclinical challenges, because they don't really show any obvious clinical symptom. We just have normal birds which are on a day-to-day basis facing these challenges and losing their energy in terms of channeling this energy towards immune activation. So now going back to the gut integrity parameter, I'll just discuss some of the alternative approaches here, and based on the lines of the previous speakers. Here you can see the epithelial cells, and the permeability can go through the tight junction as well as through the transcellular route. And with a compromised gut, we'll have reduced enterocytes with the epithelial cells, and then compromised tight junctions. So the alternative approach here I'm going to discuss are direct feed microbial or probiotics, and then phytogenic compounds. And some of the biomarkers that we'll discuss here are the epithelial cell, what happens in the epithelium, what happens in the tight junction, what happens in the mucus layer that covers the epithelium, and then the gut microbiota, and then overall how it affects the immune function, and then eventually the loss in production. Here you can see, again, this is a diagram of villi, and here is the crypt. But again, this is a trial result. Here, a control and a phytogenic feed additive and an antibiotic growth promoter. In duodenum, jejunum, and ileum, this is villus height and crypt depth ratio. So that means higher villus length and lower crypt depth. What does it mean? So basically, this length is high and this length is low if this is higher. So here, as you can see, compared to control, the phytogenic had increased ratio of this. So that provides more surface area for the villus to absorb the nutrients, and also provides more protection on the epithelium. So again, here, as I already mentioned, the phytogenic performed beyond the control and almost equivalent to the antibiotic, AGP, or above the antibiotic growth promoter. The other one that we'll discuss here is the tight junction barrier, where again, this was already discussed. This was the paracellular route, which is formed by tight junction, adherents, and desmosome and gap junction. We'll focus on the tight junction because the tight junction proteins are influenced by the actions of pathogens or endotoxins released by the gram-negative pathogens and so on. So basically, with the loss of the tight regulation, all of these can get into this blood circulation, as well as the amino acids or plasma proteins can leak back into the lumen, causing loss in nutrients. And this is an ex vivo model that that we use to measure the integrity of tight junction, where you put the mucosa here, and then this is the apical side, basolateral side, and you put a nutrient here, and then measure the permeability of that nutrient or uptake of that nutrient. On the other hand, also you can measure the resistance of the epithelium. And this is one data here. This is on birds with heat stress. The epithelial resistance. So the epithelial resistance is the current that is produced by the epithelium. The epithelium makes some resistance for the foreign objects to get through it. So the epithelium produces the current in order to avoid the entry of these molecules. So this inulin here is a macromolecule that is used as a marker to check the epithelial integrity. And the epithelial resistance, as it increases, you can see the flux of inulin goes down. So that means basically this paracellular route of transport is reduced, or it's tightly regulated, and moreover, the transcellular, which is promoted by diffusion or active uptake of nutrients, is increased. Or that's the only mode of uptake. And here you can see a multi-species probiotic compound, direct-fed microbial product, reduced the tissue conductance, or the permeability of macromolecules through the paracellular route. So this means, on the opposite side, increased the resistance of the epithelium. And this is the transport of nutrients. This is the change in the current that basically allows the active uptake of nutrients. And here you can see in the jejunum, which is the active site of absorption in the small intestine, with increased delta ISE. The next one is the mucus layer, which covers the epithelium. And why is the mucus layer is important? Because the mucus layer has two components. One is the inner part, the other one is the outer part, where the bacterial pathogens attach. And so they won't be able to translocate. And when the mucus layer gets turned over every 3 days, or because their shelf or half-life is 3 days, and the bacterial pathogen is also excreted out with the turnover of mucus layer. Here, this is an image where the concentration of Enterococcus faecium, one of the commonly used probiotic strain, and as the amount of, or the count of this bacteria is increased, the adherence of E. coli to the mucus layer, and these are two pathogenic E. coli strains, E. coli K88, AC, and MB. And as you can see, from log 10.7 to 10.9, there's a drastic reduction from 100 to almost 10% on the adherence of E. coli in the presence of the Enterococcus organism. How does it work? So basically, the mucus has attachment sites. And the Enterococcus goes and attaches in those attachment sites, and there's no any active site available for the E. coli to go and attach. So basically, the E. coli in the lumen gets excreted. If it gets attached, then it can colonize and it can cause the issues and challenge the animal. So this is the mode of action, how this probiotic can help in alleviating the challenge of this E. coli. And here you can see, again, a probiotic compound in comparison to the AGP increased the duodenal mucosal layer thickness. So the increased thickness, obviously there is a nutrient expenditure involved in that, but still it increases the protection on the epithelial layer. So this is another, the last factor here. You can see the changes in gut microbiota in the presence of a multi-species probiotic strain. This is in the water, in feed, and then antibiotic. Increased bifidobacterium with both feed and water, and increased lactobacillus count in both feed and water. So this is a cecal microflora, and then increased gram-positive cocci count with both feed and water. So basically, the presence of, or the supplementation of these probiotic strains help to modulate or influence the native microbiota, the balance of the microbiota, and change the balance towards the probiotic or good microbiota, if you will. So how these factors or the compromised intestinal integrity affects the performance? As we saw the other arrow image where it activates the immune system. So this is one of the images where I want to take your attention here. This is how the nutrients in the bloodstream is allocated to the different functions in the animal. So here you can see the brain is the topmost, has the topmost priority. If the animal is not pregnant, then the next one is the immune function. So the immune function, even though the immune system is present in the animal, when it is constantly activated, you can understand the amount of energy that goes to the immune function, then followed by visceral organs, bone, muscle, and adipose fat. So basically, we need more energy or nutrients coming here. So simply, that means eliminating this. I'm not here saying we have to produce an immune suppression. We have to be more selective in providing this immune function or activating this immune system. If we have a constant activation of this immune system, then we lose the amount of nutrients that get to the downward, and basically we lose the productivity. So here in the presence of phytogenic or supplementation of phytogenic compounds, basically reduced or inhibited the NF-kappa B target gene. So NF-kappa B is a molecule that is present in the protein synthesis pathway. So the NF-kappa B needs to be activated in order to produce pro-inflammatory cytokines, which can cause the inflammation. So if we can inhibit these NF-kappa B target genes, basically these are the genes involved, or this is a pro-inflammatory cytokine, IL-8, and ICAM1 and MCP1, and you can see a significant increase in the inhibition of these NF-kappa B target genes. And here the upregulation of NRF2 target genes, which basically plays a major role in the cell protection and inhibiting the cell apoptosis or cell death. And some of the compounds involved here, or some of the genes involved here, had higher upregulations in the presence of phytogenic, or when the body is supplemented with phytogenic feed additives. So overall, to summarize again, as I mentioned, stress on a normal day-to-day basis can increase a subclinical challenge condition, and it can compromise gut integrity and leading to constant activation of immune function and leading to drop of productivity. To wrap up, supplementation of probiotics and phytogenic feed additives may play a crucial role in alleviating or in overcoming these subclinical challenges and also to restoring the productivity. Thank you.
Thank you, Raj. Thank you. Very nice. So keep your question with you.